Okay, so yeah, hi everyone, and welcome to today's Quantum at Sun seminar. Um, so I have the pleasure of introducing Professor. If I can perhaps ask how to pronounce your name.
Oné Pagán.
So Professor Oné Pagán, who's at the Westchester University of Pennsylvania in the US, and the title of his talk is Ligand Receptor Complexities and Molecular Vibrations, Insights from Nicotine and Cotinine in Flatworms. Professor Oné Pagán, so he earned his undergraduate degree in natural sciences and MS Masters in biochemistry at the University of Puerto Rico, and his PhD in pharmacology from Cornell University. And I believe, he's currently a professor of biology at Westchester University of Pennsylvania. So without any further ado, let me hand over to Professor Pagán. So thank you again.
Well, thank you ever so much for receiving me. I'm not gonna lie, I'm intimidated to talk to a room of physicists and engineers, but I think I'm gonna give you a good show. I also want to thank very emphatically to Mr. Omid Hassasfar. I hope I said your last name right, for reaching out and inviting me and for hopefully establishing a very fruitful collaboration. So I'm going to share my screen with you so we can start the presentation. Okay, can everybody see it well? Anybody's? All right. So again, I want to reiterate my thanks and my excitement for being here with you today. And I hope we can all learn a thing or two. One of the things that you didn't hear in the introduction, of course, is a picture. This is one of the little worms that we use in lab. And yes, it has 3 eyes. We don't know how it regenerated 3 eyes, but that's only one of the many wonderful things that planarians have to offer. Also, if you let me have my coffee while we talk, I would be most appreciative. It's 8 a.m. here. Before I start, please feel free to stop me at any point if you have any questions, if you need clarifications, if you want to talk more about a particular point, please do so. Couple of things. This is an outline, a very, condensed outline of the things that we are going to talk about today. But I also wanted to give you a little more perspective of where I am from. I am Puerto Rican. If this is the North American continent, this is the Caribbean, this tiny speck here is Puerto Rico. That's where I'm from. Historically, it was a possession from Spain from like 500 years or so until the United States came. Oh, that's history. But the point is that my culture is Spanish. My first language is Spanish. My actual name, Oné, is Greek. Go figure. I have no idea. Well, I have some idea because my dad's name was Onesimus. That's from the Bible. So when his nickname was Oné, I was his firstborn. Well, here I am. So, but here in the mainland, in the US, it's a great conversation starter because people call me one. People call me Pagán. But it's not. It's one Pagán, right? It's not like that. It's Oné Pagán. All right. So, couple of things. Another thing that I want to emphasize, it's that the debt that I have to my past, present, and yes, my future students in the lab. I have been at Westchester since 2005, it's almost 20 years. And over that time, I have mentored many different students. And up there at the far left was my very first research group. Look at that brown beard that I used to have, brown and beautiful, but not anymore. The point that I'm trying to make is that those 3 young ladies nowadays are a PhD neuroscientist working in a pharmaceutical company, a nurse, and a high-flying clinical neurologist. How proud I am of those students. And as the years come by, I have mentored wonderful students. So again, it's bittersweet because they come here, they get their degree. I mentor them, I foster them. It's a little bit like a dad relationship, as a father relationship, and then they graduate and leave. The most recent iteration of the group is on the right side of the screen. This is 2024. And actually, one of them is in the audience today, Emily. So Emily, thank you for getting up early to talk to your old professor here. So the point, another point that I want to make is that everybody is welcome in the lab. I try to foster a culture where everybody's welcome, where they can learn a thing or two and hopefully have fun in the way. Also, a message to the ladies in the audience: you belong in science. Okay, you can do science, you can do math. You don't need me to tell you that, but that's one of the philosophies that I have in my laboratory. And of course, the little worms that we're going to talk about in a second. Yes, I have taught like this in class from time to time. I don't know if you're familiar with the Halloween celebration in the United States. That particular year, Halloween fell on a class day, and I went dressed as Darth Vader. They had a blast and whatnot. One of the points that I have when I'm teaching, it's that life is too short to be all grumpy. I mean, I can get grumpy like anyone, but I try to foster a lively atmosphere for a couple of reasons. First, again, you gotta be happy, okay, most of the time at least. Second, excuse me, I'm so sorry. One of the main things that I use when I'm lecturing is humor for a couple of reasons. We are all conditioned to a few minutes of close attention, followed by distractions. So what I do is that I crack a silly joke, I dress as Darth Vader, things like that. I get a courtesy laugh, pity smiles, whatever I can get, and I reset the attention of the students so they can come back to me. And from time to time, I will tell you silly jokes in this audience, which is challenging because I don't have the feedback, but I hope to entertain you today. Now, we have a specific mission in the Pagán lab, essentially to study the neurosciences from a pharmacological perspective, okay? Until very recently, we focused our efforts on abused drugs. We're talking about nicotine, cocaine, things like that. I don't have cocaine in my lab. It's under lock and key in a different building, in a different part of the building. I don't have the key. I always have to clarify these type of things, but you never know. But joke aside, semi-joke aside, we aim to discover and develop future examples of drugs that potentially could treat neurological conditions. Recently, we have begun to take advantage of the incredible ability of regeneration by planarians. We'll talk about that in a second. But along the way, as I mentioned before, I hope to offer my research students a valuable experience so they can get their feet wet in science and go on to productive careers. I am also an accidental planarian biologist. I never took a zoology course in my life, ever. My interest when I was in college and in university were more quantitative. I took genetics, I took biochemistry, I took astronomy, all these type of things. My master's in biochemistry, but then when I went for my PhD in pharmacology, I did it in a physical chemistry lab. At that time, I was studying at the biophysical level certain types of cells that express the dopamine transporter. Now, the dopamine transporter, in humans at least, is the main target of cocaine. And it's the main target that causes the behavioral effects of cocaine in humans and in mammals in general. Now, one fine day when I was doing my PhD, the paper that is showing at the lower part of the screen appeared from a group in Philadelphia, United States. I got excuse me, very enthusiastic about it. And I went to my advisor and I said, we have to use these worms for our experiments. And well, he said no. I understand. He was a hardcore physical chemist. Okay, animals, come on, and that, you understand what I mean. He actually told me, when you get to have your own research group, you do that. And that's precisely what I did. When I was hired at Westchester University, I educated myself. Again, as I told you, never took a zoology course in my life. But I began reading about planarians, educated myself in that, and I was eventually able to integrate the pharmacological sciences, now regeneration, and this type of endeavors with planarians. Am I the first person to think about in those terms? No, but it didn't get into fashion, quote unquote, until relatively recent. Now, what are planarians after all? Planarians mean literally flatworms. Flatworms, like a little ribbon, if you will, something, I don't know, like a cell phone or something like that. There are three main general types of planarians: marine planarians, terrestrial planarians, which are agricultural pests because they are predators of earthworms. So that's a big problem in certain places. And freshwater planarians, those are the ones that we used in my laboratory. The picture at the upper right shows an image, and I realized that it's not the same type of coin. This is the same scent that I'm showing you in the picture is something like this, okay? And a planarian on top of it, okay? So they are relatively small organisms. They're good to work with because they cannot bite you. They will not fly and poison you, nothing like that, okay? They are relatively inexpensive to buy and maintain. We don't need a special incubator, nothing like that. But they have the quote-unquote first example of a vertebrate style brain for a variety of reasons, just not only structurally, but biophysically and biochemically. They display pretty much the same type of neurotransmitter systems as vertebrates do. Okay, so they are poised to be developed as an interesting animal model. Prior to the renaissance of planarians in pharmacology, as it were, they have been used and still are very much used as an animal model in developmental biology and in pharmacology. Why? Because they are one of the few organisms that actually can regenerate virtually any body part that they can that we can cut off, including their brain. I'll elaborate that in a moment. But at the lower part of the picture, courtesy of a friend of mine in Germany, there is a series of micrographs of the same planarian, following since we cut their heads off until they recover their eyes. I'm going to show you large pictures of the planarians in a second. This type of planarians, they regenerate a whole brain within a week. Nervous systems are notoriously famous or infamous, I should say, because they cannot heal easily. Think about, sadly, a person with brain damage from a car accident, okay, or spinal cord damage from an accident, Alzheimer's disease, Parkinson's, anything that can go wrong neurologically. Imagine if we learn how to recover nervous system's body parts correctly, because it's not uncontrolled cell growth. That's cancer. We don't want cancer, okay? And planarians are not the only organisms that can regenerate, but they are one of the best ones at it, as it were. Now, a couple of things. I work at a predominantly teaching institution. We offer bachelor's and master's. That being said, we take research very seriously. In the years that I have been at Westchester, I've had close to 100 research students. Some stay for a couple of months and then they leave. Some stay for years and complete a degree and papers and whatnot. We have published about 27 papers or so, with about 20 more in several developmental stages, abstracts, presentations. As part of my scholarship, I have written 3 books, and I know a little bit or two about planarians because I wrote the first and only book about the neuroscience and pharmacology of planarians from Oxford University Press. And I'm not saying this to be, I don't know, to be arrogant or anything like that. It's just to let you know that I know what I'm talking about. But moreover, I love what I'm talking about. It's like all of us, when we were kids, we went to ponds and collect tadpoles and worms and whatnot. Many people outgrew these type of things. I never did. So I'm having the time of my life, as it were, right? So in addition to that, I am engaged in science communication for the public in the form of blogs, podcasts, all these type of things. And if we have time, we can elaborate on that later on. But let's go back to the science. Now, remember that when I told you that we can cut planarians in pretty much several pieces, and each of them will regenerate a complete worm, full regeneration. At left, there's a series of decapitated heads. Don't be afraid about, scared when you hear the word decapitated. Whoops, sorry, that's not what I meant to do. Because the heads are completely alive. They will swim happily, and eventually those heads will regenerate a whole body. But if you have your body, a decapitated planarian body, don't worry, it's alive still. And the body will eventually regenerate a head, okay? And there's a lot of implications and about this type of things, okay? One of the most common questions that people ask me is, if you got 2 planarians and each of those regenerate a full body, are they clones? Yeah, they are like non-molecular biology generated clones because they are essentially identical organisms with the same genome. Sometimes they do weird things like regenerating heads in opposite sides of the body. This is another example of that. This particular planarian is tiny. It's about a millimeter long. And remember, a millimeter is about the width of a fingernail, something like that. And this particular worm was cut in like a little rectangle, and each of the ends regenerated a head. We can manipulate the appearance of heads in weird places in planarians genetically, or by manipulating the bioelectrical environment of the planarians in the cell. And we are beginning to do experiments in that respect. Are we okay? Do we have any questions, any comments? Am I good to keep going?
Can I ask a question quickly? Sorry.
Absolutely. Don't apologize for questions.
I was just wondering, when you have a body that regenerates a head, does it retain sort of any of the behavior? Does it retain anything that the brain had before?
Yes, most of it. There were experiments in the 1960s which were controversial, in which a researcher, he would train planarians in a classical conditioning way, as in an experiment with any other animals. So he would then cut the heads off, took the heads away, and when the bodies regenerated their heads, a significant proportion of the worms retained the memory of the training that they did with the maze or conditioning or whatever it was. At the time, it was understandably controversial. People didn't believe it and whatever, and that kind of died away. But more recently, in the less than maybe 10 years ago, another researcher with better controls, computerized visualization of behavior so that will eliminate bias and things like that, replicated those experiments. That brings a question: where are memories stored in the worm? And if we can get cocky, we can keep going. Where are memories stored in a particular brain, for example? Of course, even though they are similar to us, worms are undeniably simpler. Okay, so, and the recover of memories is not perfect. As I said, a significant portion of them retain their memories, okay, but not all of them. So that gives us, yes, go ahead.
Did they find in the worm's case where, do they have any idea where the memories were stored? In the bioelectric and the ion channels, or?
Well, they hypothesized that And that's a hypothesis that needs to be carefully studied. I have seen no studies about that yet. That is stored in messenger RNA molecules. I don't know exactly what that means. That's the only correlation, because sometimes you can get, for example, you can train the planarians, isolate messenger RNA. Let's just say RNA for short. And then if you inject that RNA to naive planarians, they kind of gain the memory of navigating a maze and whatnot. I know it's crazy. From the perspective of a biologist, a scientist, it's hard to understand how it works, but it happens. And it's undeniably a fascinating area of research.
Thanks so much. That's absolutely fascinating.
It is. Did I answer your question, more or less?
Yes, you did. Thank you very much.
Okay. So then planarians display a variety of behaviors that can be compared to behaviors of people who get addicted to drugs, sadly. And again, drug addiction is a horrible problem. I'm not making any fun of it or anything like that. But if we understand the basis of addiction and all this type of phenomena in humans, that can help relieve some human suffering. But for example, when planarians are given an abused drug, let's say nicotine, okay? You can put nicotine in the water, leave it for a certain period of time. Of course, you have to be careful about gauging the concentration so it will not be toxic. You leave the nicotine there, then you take it away. The planarians go crazy. As you can see in the left, they go like head bopping, they corkscrew, they swim around like crazy, they shake, pretty much as in a person who quits drugs, or even, I don't know, tobacco for that matter, and they go into what we call withdrawal. So this phenomena in planarian, these movements in the left, we can consider those surrogates for withdrawal symptoms in humans. Okay, so that will entail a chronic administration, not toxic administration. But then we can have seizure-like movements that we use to assess toxicity in certain compounds. For example, and I will show you a picture in a second. When the planarian A, just in normal water, and this one, I don't know if you can see it, that it's like curved in like a C-like fashion. It's undergoing a seizure right now. Now, on the top, we have two planarians here right now. They are in water, okay? And in a second, you're going to see a plastic pipette. I'm holding the pipette, by the way, and I'm going to administer the planarian a really high concentration of nicotine. We're talking millimolar amounts. Here comes the pipette, and in a second, you're going to see the planarian scrunch as the water gets, there you go. And in a few seconds, you can compare it to the planarian on top. The lower one, it starts seizing like crazy. It's twitching. We can quantify those seizure-like movements, and we can assess the relative toxicity of whatever compound that we want as a function of concentration. Truth be told, we all feel very sorry about the work in killing the planarian, giving them toxic compounds, but it's way better than rats, and God forbid, better than monkeys or any big animals, okay? Animal research is necessary, but we can choose what animals to work with. Does that make sense? I hope it does. Now, based on a series of experiments from our laboratory, this, before I forget, this is another type of movement that we quantify, and this is going to be relevant for the experiments that I'm going to describe. We call that simply measuring motility. Right now, they glide at a certain speed, and we put the dish where the plate is at over a grid line, a 1 centimeter line. Actually, we are working to automate this process through computer visualization and things like that to expedite experiments. So I'm very excited about that. But we can quantify those that movement, and we can add compounds at a variety of concentrations that will either slow down or speed up the planarians, for example. If you give them small concentrations of caffeine, they speed up. the planarians, just like caffeine perks people up. So it's that similar, their nervous system functioning. Now, again, essentially, we do the pharmacology of flatworms. That doesn't sound very glamorous or anything like that, but it's what we do, and it's incredibly interesting. Well, some of the experiments that I'm going to show you in a moment are focused on nicotine. And of course, nicotine is an abused drug that it's a problem worldwide, right? And that is directly to a variety of conditions and whatnot. But let's stop here for a second and ask ourselves the question: why a tobacco plant would make nicotine? Okay, so nicotine is not used in the metabolism of tobacco plants for nothing, absolutely nothing. So the current thinking is that in evolutionary terms, nicotine and related compounds that we will talk about in a second are, in evolutionary terms, the first pesticides. They are natural insecticides. You see, if we have like a tobacco plant here, a bug goes there and starts nibbling on it, it ingests a certain amount of nicotine, the bug dies, okay? But a person who smokes tobacco will get more or less the same concentrations of nicotine in their bodies. We are much bigger animals, but we have similar nervous systems. The same amounts that will kill an insect will cause psychoactive effects in us. The same goes for cocaine in coca plants, morphine from poppy plants. Those compounds, they do nothing for their metabolism of the plant where they are generated. So these type of compounds, again, are considered the first insecticides, natural insecticides. And it just so happens that since we share an evolutionary relationship with insects in terms of receptors and whatnot, and many other things, again, the amounts that will kill a pest will intoxicate us in the pharmacological sense. Now, most of the talk, scientific part of the talk that we are going to talk about in a second is based on a hypothesis paper that I was fortunate to publish very recently. And I will tell you the story about how I got into quote unquote quantum biology. If I was an accidental planarian biologist, I am a hyper accidental quantum biologist, as it were. This was very recent. And again, please keep in mind that this is a hypothesis that we are beginning to, including our collaboration with Omid, we are beginning to try to get evidence for these type of things. But the circumstantial evidence is very compelling. Now, for that, let me give you a very short course on receptor biology. When we talk about a ligand, we are talking about a compound. We can call it a drug. We can call it a toxin. We can call it again, a compound, a chemical, okay? But in pharmacological parlance, it's a ligand that binds to a specific receptor. If you look at the right side of the screen, I have a representation of a receptor molecule with a part that's labeled as being an active site. An active site is a three-dimensional shape in the protein receptor molecule that is complementary to the shape of the ligand. And the current understanding of the ligand-receptor interaction is precisely that, the lock and key hypothesis, in which the three-dimensional shape of the binding pocket is complementary to the three-dimensional shape of the ligand, okay? Just like a lock and a key. A variation of the lock and key idea is the induced fit hypothesis, in which we have, again, we need the three-dimensional shape, but we need a little click to put it in place. As if you were playing with a toy like a Lego piece, that it's not enough for them to have complementary shapes. You need to push it a little bit for the toy to be assembled. Exactly the same type of thing. But that's not the whole story, because we need to understand thermodynamic considerations, okay? A variety of other concepts, including lately quantum mechanics, okay? Come on. Now, in this context, here's just a schematic view using the same nomenclature. Again, ligand is the drug, receptor is a protein that accepts that drug. When we have physiological binding, when the ligand binds to the drug, it triggers a conformational change in the protein to do something. It could be a second messenger, it could be an ion channel, it could be enzymatic interaction, enzymatic reactions, whatever. Schematically, we show it as receptor plus ligand, okay? This complex, ligand-receptor complex, it's what is the active form of the protein. These two arrows indicate that it's a reversible process. Physiologically, it's reversible. There are some compounds that can kill an enzyme by covalently bind to them. Then, in that case, we're not talking about a drug, we're talking about a toxin, okay, or a poison. And all the pharmacological equations that I'm going to show you, they will not apply under covalent circumstances. We're talking about reversible binding. Now, sometimes we can have competitors, meaning that if we have a compound, look at the middle panel here, a compound that binds to the same binding pocket as the ligand, but does not induce a conformational change, while at the same time preventing the binding of the ligand, we are talking that a competitive inhibitor, okay? Meaning that this is the binding of the two is mutually exclusive. It one's or the other. One binds or the other. But then the inhibitor, by definition, does not trigger the conformational change that is necessary for a protein to function. I hope it makes sense. Can you everybody give me a thumbs up if it makes sense? Okay, here we go. Thank you for this. I work much better with feedback. Then, let me give you an example of competitive inhibition. If I have my phone here that has a shape that is more or less complementary to my hand, right? But if I want, with the same hand, grab my stapler, okay, I cannot grab my phone. And I can certainly, unless I am very disturbed, I will not even try to make a call with my stapler, okay? That's the main idea of competitive inhibition. It's one or the other, but one of them does not cause the desired effect, okay? Then there's the idea of non-competitive inhibition, in which we have a compound at the lower part of the panel, in which it binds to an alternate place in the molecule, let's say here, okay, closing the binding pocket. It's not interacting directly with the binding pocket, but it closes it nonetheless, so I cannot grab my phone, as it were. So there are many variations of the theme, okay? So because nature doesn't care about how we decide to divide it or describe it. But in pharmacological terms, these are the three main ligand-receptor interactions: competitive inhibition, non-competitive inhibition, and physiological binding. This will become relevant for what we are going to talk about.
Can I ask one more question? Sorry.
Yes, no, please, don't apologize. Thank you.
Is the non-competitive binding like an allosteric effect, or?
That's exactly right. The non-competitive bind is considered allosteric because it binds to a place different from the binding, the normal binding site. When we're talking about competitive inhibition, on the other hand, is steric. Okay, steric, same site. Allosteric, different size. And thank you, because this will become very relevant for what we are going to talk about in a few minutes. Thank you, thank you. I love it. Because despite what you may think about professors, at least I don't love to hear myself talk. I like the conversation. All right? Now, the mystery about this is, particularly in the case of competitive inhibitors, if we have one binding pocket and two compounds bind to it with different physiologies, what's the key, the structural key? It must be structural, we think, about the binding of one compound that induces a conformational change in the receptor versus another compound that does not induce that conformational change or induces it partially, for example. One of the best known examples is the case of morphine and related compounds. Morphine is one of these compounds that is a natural pesticide from plants, but it's also a painkiller. And it's a very good one, but of course, as with any medication, it has certain undesirable side effects, including the possibility of addiction. Now, with relatively small changes in the molecule, as simple as acetylation, okay? Look at the lower part of the picture, heroin, which is a much more potent compound than morphine, and it's the cause of many fatal overdoses. And of course, that's undeniably a big problem, okay? It's also a painkiller and whatnot, but it's too good, if you know what I mean, with, again, the bigger possibility for, extreme toxicity and death. Now, then we have the example of the compound here, naloxone, which is very similar to either morphine or heroin, but with certain structural differences. It binds to the same site, and it acts as the other two do, and it acts as a competitive inhibitor of the two. It's actually an antidote. When a person gets an overdose of heroin, in an emergency situation, what they give the person is naloxone, and it wakes up the patient like this. Okay, it's almost a resurrection, if you know what I mean. So, why the main pharmacological problem is that why compounds with very similar structures, and as you will see momentarily, molecules which may differ from each other in one atom can have very different pharmacological properties. That's what we gain, another area of research courtesy of planarians. Before going into that, thank you again for letting me have my coffee. We need to go into that certain very preliminary aspects of neuromuscular transmission, because we need to understand the receptor that we are going to talk about. If I have to move my hand and make my muscles contract and whatnot, the nerve sends a signal to the muscle by releasing neurotransmitter molecules like that. In normal muscle, that neurotransmitter is called acetylcholine. It's probably the first neurotransmitter that it was discovered, has a very rich history. But the thing about it is that the receptor that it binds to, they also bind another compound called nicotine that we have alluded to before. Therefore, the whole class of receptors, as I will show you momentarily, are collectively called nicotinic acetylcholine receptors. They are acetylcholine receptors that are sensitive to nicotine. The really interesting part, the more interesting part about this from our perspective, is that there are varieties of nicotinic acetylcholine receptors in the central nervous system of humans. That's where nicotine acts to induce their pharmacological effects. When you smoke a cigarette, and I know the numbers because I've read about them for another project recently, in about 6 seconds, enough nicotine gets to your brain to induce their pharmacological effects. There's hundreds, maybe thousands of different types of nicotinic acetylcholine receptors in the central nervous system. And we assume that those are the receptors that are going to be affected by nicotine in our flatworms. And genomically, we know that there are genes that are analogous to nicotinic acetylcholine receptors. And by the way, from now on, I'm going to call them nicotinic receptors for short, but please keep in mind that their natural ligand is acetylcholine, They look more or less like this. They are pentameric proteins. If you look at them from above, it looks like your 5 fingers here. And it has 2 binding sites for nicotine and for acetylcholine, naturally, but then for nicotine. Once 2 molecules of nicotine acetylcholine bind to it, it opens a channel, allowing positive, I almost touched my screen. You don't see what I'm touching, but sorry about that. So we have the positively charged ions that flow through the membrane to induce any biophysical phenomenon. Why do we know so much about nicotine receptors? There's a couple of lines of evidence that we used to do that, not we as in my lab, we as in the scientific community. Highly electric organisms like electric rays, they display a modified muscle tissue, they call it electric organ, in which they produce nicotinic receptors at 1,000-fold concentration as if we have it in our muscles here. And their cells are also, the electric organ cells are organized like a battery. When that organism activates their nicotinic receptors, it can give you a jolt of about 400 volts, something like that. And that's a big jolt. But because we can isolate receptors from these type of organisms biochemically, we know so much about these particular proteins. But I digress. Let's keep going. The main physiology of chemical neurotransmission has two schools of thought. One is conformational selection and conformational induction. In reality, I'm convinced that there's going to be a combination of the two. All right, so, but just for to understand it better, let's talk about them separately. Conformational induction means that when we have a receptor here, it remains closed until the ligand, in this case acetylcholine, binds to the two sides of the receptor, triggering the conformational change, and opening the channel to allow for ion flow, okay? And again, these nicotinic receptors are related to neurological diseases, addiction, conditions like epilepsy, okay, memory, all these type of things. So they're undeniably important. But then we have the idea of conformational induction, okay? All receptors, regardless of whether they are ligand-gated ion channels like the ones we're discussing, or metabotropic receptors, second messengers and whatnot, they exist in three basic conformations: resting, okay, active, and desensitized. In the resting conformation, the receptor is, of course, resting, closed, but amenable to receive the signals from the neurotransmitter, all right? So right now is closed. When the neurotransmitter binds, we trigger a conformational change to the active receptor. So we have the two conformations there. But also physiological, even in the presence of receptors, the receptor goes into a state that is called desensitized, because it's like driving a car with a gasoline pedal, a petrol pedal, but with no brakes. If we cannot brake, the car can take us to one place, but it's gonna be problematic. So physiologically, the desensitized state is the brakes, quote unquote, of the receptor process, okay? Once the channel becomes desensitized, it stays for like that for a certain period of time, and it cycles back to the resting conformation, which is sensitive to the neurotransmitter. There are intermediate conformations between those three, for all intents and purposes, infinite. And we could pharmacologically dissect all of them. But these three are sufficient to explain most pharmacological phenomena, okay? But then keep in mind that in an organism, we cannot work only with one receptor, right? We work with thousands, perhaps millions of molecules, maybe billions in a molecule. That's where the idea of conformational selection comes in. In that idea, it states that in a normal tissue, there will be more or less an equal concentration of each of the three receptor conformations: resting, resting, active, and desensitized, right? And they are kept like that due to in that conformation due to thermodynamic considerations. That's what I was trying to, the word that I was trying to find. In the presence of agonist, okay, an agonist is a compound that activates a receptor. That agonist present shifts the population of the receptor towards preferentially the active side. So there may be some receptors that need more agonists or they remain inactive, but statistically, most of the population will remain active under the presence of agonists until they get desensitized. The idea is that then, in the presence of an antagonist, we are shifting that equilibrium towards the closed state of the receptor, regardless of whether we are talking desensitized or resting. It stays closed. So, and again, it's essentially a distillation of the same idea. The receptor tends to remain inactive until we trigger it with a ligand, a compound, okay? But in reality, the conformational selection can also be true because we are dealing with a particular tissue, and in our case, a whole organism with everything that it entails. Still good? You need your coffee too? Okay. Now, the paper that started it all was in 2016, in which we studied nicotine and a metabolite of nicotine. Actually, the biggest the most common metabolite of nicotine in humans is called cotinine. Metabolism is in the context of pharmacological metabolism, they are processes that modify the drug for them to be excreted because the body recognizes them as exogenous molecules, okay? So they detoxify compounds by metabolizing it so they can be more easily released in urine and all these type of things. So in 2016, this paper, led by an undergraduate student, then described that cotinine, and I will show you a structure of cotinine in a moment, which is virtually identical to nicotine, counteracted not only the acute administration effects that mimic toxicity, but also the chronic administration effects in planarians, these, of course, that mimic addiction. And just to show off a little bit, where are these people today? Dan is a medical student. Matt is an actual dentist. Deborah is a college instructor right now. Sean is a pharmaceutical scientist. And Erica is a physician. I think she's a surgeon. Again, this is a sense of immense pride when my students get what they want in life. But let's continue with the idea. One of the effects of cotinine against nicotine is the prevention of the expression of the withdrawal-like movements that I showed you before, with the exception of these two, corkscrew and tail twist. They still did that in the presence of cotinine. That suggests different neuronal pathways for both, but we can study that later in my laboratory. But the main point, cotinine seems to be an antidote, if you will, against nicotine in planarians, and there's some evidence that that is acting like that in mammals too, as it were. Now, I love this quote because you know this by heart. When in primary school, they tell you that the scientific method is make an observation, formulate a hypothesis, and then do. No, it works like it works by observation, but like, this is interesting. This is funny, and that's why we got this contrasting activities between nicotine and cotinine caught our attention. Please keep in mind that the only difference between the two is this area here, in which this CH2 carbon atom is substituted by a carbonyl group here. Minimal changes. Then why does that make nicotine an activator, quote unquote, and cotinine an inhibitor, as it were? Of course.
Can I ask another question?
Go ahead.
Do they have very different vibrational spectra, or?
That's one thing that I'm going to mention that, and actually the short answer, yes. Yes,
Okay.
Omid did those, and we were discussing this yesterday. Yeah, we
Thanks.
don't know exactly what it means because there's a question about certain aspects of the charge status of this nitrogen here and this
Okay.
nitrogen there. But because charge atoms will change the spectrum. And in physiology, there may be multiple physiological states. There may be multiple states of the molecule, which may confound this. You ask the best questions. Use your name, Bethany?
Yes, thanks.
Okay,
Yeah.
thank you.
Thanks.
And I'm sure you all ask wonderful questions, but I love questions when I'm chatting and in lecture and whatnot. So yeah, thank you. So the spectra are different. We think that's what's going on in terms of quantum tunneling, electron tunneling and whatnot, but we haven't proven that yet or gathered evidence about it. There are two possibilities about the relationship between cotinine and nicotine. One of them, that they are competitive inhibitors. Remember, one binds to the same place as the other, okay? Or we have allosteric interactions in which presumably cotinine is binding to a place that's preventing the conformational change or kicking nicotine out of it. We don't know. Those are the two possibilities. From our perspective, the one that would be more consistent with what we're talking about is this one, that they are binding to the same site, but we don't know that yet, okay? We have evidence that indicates that they are binding to the same site under certain conditions, okay? And I'll show you the experiments in a moment. This is related to the idea of the concept of the principle of microscopic reversibility. Some of you may have heard this in the context of when Einstein worked with the Planck radiation equations. He used this principle to deduce this type of ideas. Essentially, it states that any individual process, for one individual process, there is a reverse process in a state of equilibrium. Remember that we're not talking about covalent interactions. We're talking about reversible interactions. And the average rate of one process would be very close to the average rate of the other. They are concordant. They are not independent. They are proportional to each other. Okay? Then this concept has been proven valuable and applicable to enzymatic reactions, receptor-ligand interactions, as we will talk about in a second, and many other phenomena, okay? Now, my research advisor at Cornell, and I was talking to Omid yesterday, I wish he were alive right now. He died a few years ago. He was in his 90s. His mind was completely clear. He was a good guy. But he would have loved this idea of quantum biology. He would have told me that I'm wrong, but he would enjoy this. He came up with the idea that certain compounds that may act as inhibitors of others, perhaps by metabolic transformation or through some other considerations, relate to each other in a receptor due to the principle of microscopic reversibility. This will assume allosteric interactions, okay? This is in direct, contradiction to the hypothesis that I'm presenting for nicotine and cotinine. But this is where I got the idea of applying it to nicotine and cotinine. You'll see why. You see why. We have in this scheme, okay, let me see. Okay, a receptor binds to a ligand, and A stands for allosteric modulator, whatever that is. So let's assume that we're talking about a nicotinic receptor. So we need two ligands to bind to the receptor. This is resting, a rapid conformational change that opens the channel. Okay, so let's only look at the uppermost scheme. Receptor, ligand, binding, conformational change, opening of the channel. Okay, so that's it. But if we have an allosteric site, okay, that binds something, right? We can differentiate whether we are talking about an activator of the receptor that does not necessarily have to be the normal ligand or an inhibitor of the compound. For example, if that activator binds to an allosteric site that stabilizes the open or active conformation of the receptor, we are in this place and we call it an activator, okay? But if we have a compound that acts as an inhibitor of the process by stabilizing the closed conformation of the receptor, we call it an inhibitor, okay? So this is an expansion of the original agonist-antagonist modulator concept in pharmacology, okay? He published this almost 25 years ago. In this particular example, he studied cocaine analogs. Even though cocaine is not the main target for nicotinic receptors, at very high concentrations, it can bind to it. And that's one of the things that he decided to study. So he hypothesized that metabolites of cocaine, like these two, when a person ingests cocaine for a variety of reasons, and it's being metabolized by the body, these are the two main metabolites, okay? He hypothesized that metabolites of the original compound will act as allosteric inhibitors of the effect of cocaine in this particular receptor. And it did work. In the paper this previous paper, he described this particular effect. So that gave evidence for the idea of those allosteric sites. And then when I started thinking about cotinine after 2016 and whatnot, I extended this idea to nicotine, cotinine, and other analogs. And I published it in a chapter on a book about nicotine. But at that time, I was not even thinking about quantum biology. I was thinking about this particular idea. So one of the things that I love the most about science is that an ostensibly simple observation can lead to so many things. And this is one of the examples. Now, from now on, I'm going to show you planarian motility experiments and our interpretation of those, confirming and extending the idea that cotinine, it's an inhibitor of nicotine. Am I still good on time? I know that I'm at the 1 hour mark.
Yeah, I think we can just continue. Sorry,
I got a lot of feedback. If I'm still good on time, can you give me a thumbs up?
I'm just not going to talk.
Yeah, I just want to be respectful of your time because I can talk about this. Yeah.
Okay. Sorry, we had like a lot of feedback issues this side.
Okay.
Yeah, you're fine for time. Yeah, sorry about the feedback.
Okay, no, that's fine. It's just that again, I don't want to abuse your kindness. But even though I'm enjoying this, and I hope you're enjoying it too, but I want to be respectful. Anyway, so let's talk about motility experiments. Okay, what we do, very simple. We put a planarian on a dish, on a plate, and then we count how many squares it crosses, okay, each minute for a period of, I don't know, up to 8, 10 minutes, whatever the design calls for. If in the first minute it crosses 10 squares, the cumulative crosses is 10. Okay, we're going to add them up at the end. You will see where we are getting with this. In the second minute, it crossed 8 more. So the cumulative is 18. You know where I'm going with this. If it crosses 11, the cumulative is 29, and so on and so forth. The point is, if we plot the cumulative crosses as a function, of time, we get a straight line. Okay? And since we get a straight line, it's beautiful because we can calculate the slope and we can start to add some mathematical sophistication to the analysis. Okay? A typical planarian has a slope of between 8 to 10 squares per minute, something like that. So that's a typical planarian movement. A compound that slows down the planarian will display a reduced slope, okay? A compound that speeds up the planarians will display an increased slope from the control. As I told you before, low concentrations of caffeine speed up the planarians. And as you will see momentarily, low concentrations of nicotine also speed up the planarians. But high concentrations of nicotine is essentially toxic to them, slows them down. This is an experimental idea that was published in 2001, but we modified it extensively to add them I don't know, the mathematical analysis, all these type of things. Now, if we get a family of slopes, all right, and normalize it to the control slope, we get a concentration response curve or a dose response curve, which is what I'm showing you next. These are some of the experiments in our lab in which we added nicotine at increasing concentrations, showing that it slows down the planarians. For example, in this scale on the y-axis, 1 represents 100% of the motility, 0.5, 50% of the motility, and so on and so forth. So at low concentrations of nicotine, this is a log scale, by the way. So I'm a biologist, but I know a little math. Stupid joke. Okay. So at very low concentrations of nicotine, it's very close to the control, 100% of movement. As we increase the concentration of nicotine, it slows down the planarian until at high enough concentrations, the movement is completely abolished. Okay? We get a parameter, just an arbitrary parameter, that tells us the concentration that we need to add the worms to slow them down by 50%, give or take. In this particular series of experiment, it's about 114 micromolar, which is consistent with everything else that we have published before. And we are using that point, okay, that reference point for our further experiments, okay? Now, we established here, as in previous experiments, that nicotine at high concentrations slows down the planarian movement, all right? But then cotinine here in blue does not abolish planarian movement at very high concentrations. We're talking 3,000 micromolar, 3 millimolar, something like that. And we know that because we fit this equation to a line, the slope is statistically no different from 0. It's a straight line, showing a non-concentration dependence of cotinine for planarian's reduction of movement. Another interesting thing is that low concentrations of nicotine, they seem to increase the planarian speed. And we know that because the y-intercept is about 1.2, about 20% more than control, and the line is not differently statistically. The slope is not statistically different from 0. So then again, it's a big plateau, over 20% plateau, something like that. So that tells us that at low concentrations, nicotine speed up planarians, okay? At high concentrations, nicotine slows down planarians, maybe at different binding sites, as we will see momentarily, and cotinine does nothing, apparently nothing, at those high concentrations. Make sense? Okay, so then we use the reference point of the 100 micromolar nicotine to do a series of paired experiments in which we added nicotine at about the concentration that causes reduction by 50%, give or take, and a pair experiment with the same concentration of nicotine, but a certain concentration of cotinine. Okay, so at this in this experiment, nicotine at more or less 100 micromolar reduces motility by about 50%, but the presence of cotinine takes it almost all the way back to control. in this experiment, nicotine at the same concentration, more or less the same inhibition of movement, cotinine at a higher concentration, all the way to control. Same thing with all experiments. In red, nicotine reduces motility, and in blue, the combination of cotinine with nicotine rescues that motility decrease with a certain degree of dispersion. That's expected, especially in a biological experiment, in which we are doing behavior, in which we are doing interacting with millions of receptors in that type of experiment. Moreover, when we plot, it's like a normalization of the effect of nicotine over the combination, we get a positive slope indicating concentration dependence. Okay, and we do the statistics, it's significant and whatnot. So the higher we go on cotinine, the higher we go in the alleviation with some dispersion. And we need to add experiments to that. And in fact, we got the results from a submission of this work. One of the critiques of the reviewers, we need more data, and they are absolutely right. And we are working on that. Then we did a series of experiments of a low concentration of nicotine, about 50 micromolar, which significantly increases the motility of the worm, but cotinine doesn't see that, doesn't alleviate that increase in motility. Okay, so it's essentially, it's not seeing it, as it were. What we're thinking about is that we are seeing two different sites, as it were, because we did the statistics and everything. The point that I'm trying to make is that, what do we have so far? Nicotine decreases planarian motility, cotinine does not. Cotinine alleviates the motility reduction. Nicotine at low concentrations increases speed, and it doesn't seem to alleviate that increase in speed. But we have to analyze the data to be more sure, because we have some dispersion and whatnot. We are doing several types of experiments, including something called child analysis. I don't know how it's pronounced. I was debating with Omid this yesterday. I don't know how to pronounce it, but it's written like this, that will tell us whether nicotine and cotinine are acting sterically or allosterically, depending on the condition. And those are ongoing experiments, and wish us luck. I mean, it's the best thing. But then, when we examine the structures, again, of nicotine and cotinine, they are virtually identical. Oh, and by the way, I already, a couple of days ago, I received this compound is yet another analog of nicotine that we are eager to test. It's called nornicotine, and I don't know what's going to happen. Nobody has done it before. So am I hypothesizing that it will act like nicotine or cotinine? I don't have enough information either way. We're going to test it. Now, in addition to this, we can do molecular modeling experiments. Well, we can do so many things that it's incredibly interesting. And the way physiologically in which nicotine and acetylcholine act seems to be consistent with electron tunneling due to a series of circumstantial pieces of evidence. At left, this is the nicotinic acetylcholine receptor, again, the pentamer that I showed you before. Its two binding sites are made out of specific protein loops, peptide loops, of two different subunits of the receptor. This is a representation of an acetylcholine molecule. This represents a very rare series of vicinal disulfides. You know the disulfide bonds? These are two next to each other cytosines that bind together to make a disulfide bond. Without this bond, nicotine does not act, or acetylcholine. The positively charged part of nicotine is related to loop A, and in loop A, there's a series, depending on the species, there are a series of aromatic amino acid residues, in this case is tryptophan, that displace, due to their benzene ring, a dual partial charge nature. At the center of the molecule, maybe let's go to this lower part. At the center of the molecule, we see a partial negative charge, and at the periphery of it, in the carbon atoms, a partially positive series of charges. The current understanding is that the positively charged milieu of nicotine and or acetylcholine interacts with the partially negative charge of the tryptophan and other aromatic molecules to interact with each other and cause a conformational change and everything. But based on the idea of electron tunneling that we are hoping to explore, we found that aromatic molecules, they are capable of electron tunneling in a variety of contexts. Nobody has ever related these type of things to nicotinic acetylcholine receptors or the general family of ligand-gated ion channels. Then, good, my computer froze. Bear with me for a second. Oh, don't do this to me. If you'll excuse me for a second, I'm going to close this and trying to open it again. Electronics, gotta love them. Okay, can you see the screen now? Good? Okay, so, and again, I apologize for this. One of the questions that I personally ask myself consistently is, what if? What if? And I began to, for a series of historical circumstances, I began to think about quantum biology in this context. This is a quote from one of the people who got me into that, Professor Clarixi Aiello. She's the person who runs the quantum biology chats, the weekly quantum biology chats. Everything born quantum dies classical, okay? And I love that quote. I'm sure that that I don't quite understand it in the context that she's saying it, but I love it. Here's the deal. A few years ago, I read a book that most likely all of us have read. I'm rereading it right now, refreshing myself. Life on the Edge, from one of the two pioneers of this idea of quantum biology, Drs. McFadden and Al-Khalibi. Then I have a planarian friend. She's another person who works with planarian, Dr. Wendy Bean. We were talking about planarians, about some other, in other context, and she mentioned in passing that she was in a group about quantum biology. And I asked her, well, what's that? And I have read the book, but I thought nothing of it. And I told Wendy, if you ever go to a meeting, let me carry your books. I would love to get involved. So she put me in touch with Clarixi. I got into the list, and I began interacting with the community that led eventually to the hypothesis paper that I mentioned. All of this came about from the pioneering work of Dr. Luca Turin, who in the 1990s proposed an albeit controversial hypothesis. And to say that it was controversial is an understatement. In my view, he was mistreated by segments of the scientific community, in which he proposed that olfactory senses are spectroscopic in nature, in a way similar to audition, which work with frequencies and wavelengths as light too. He proposed that olfactory receptors, which are G-protein receptors, second messenger receptors, act by vibrational properties of the ligands interacting with the receptors due to a specific type of electron tunneling called inelastic electron tunneling. I know you know all about this. I'm not going to try to explain it, so I don't want to sound ridiculous. So, but you know what I'm talking about. He states correctly that the lock and key hypothesis is not enough to fully explain the ligand receptor interactions in this context. Okay, so he proposed again that the mechanism through which odorants interact with GPCRs is inelastic electron tunneling. And why did he propose that? Humans, for example, we have 300 or so olfactory receptors, but then there's millions of different smells that we can detect. And let's not even go to dogs, for example, which can detect even more. They're more sensitive to that. So this is consistent, again, with smell being an spectroscopic sense, okay? Same idea that two virtually identical compounds can smell differently, okay? Implies, again, the concept that the receptors are seeing the molecules differently somehow, and maybe structure is not enough. But then again, quantum electron quantum tunneling. This was expanded a few years after by Dr. Jennifer Brooks' work with the swipe card model of odorant recognition. Essentially, the way I understood it, and Omid liked it, so I think I must be more or less on the right place. If you get your driver's license, which is more or less the same shape as a credit card, you cannot go to pay for breakfast with your driver's license. You need a credit card with a specific electronic and magnetic configuration that can be recognized by the machinery, and you can pay for your breakfast, okay? So this is the same idea, that the three-dimensional structure of a ligand, it's necessary but not sufficient to account for whatever properties that we need to explore, okay, for the effect of the receptor. I've been eagerly waiting for a continuation of Dr. Turin's work, and yeah, we found a preprint that was, I think it was 2021. Yeah, 2021. I don't know why it's not published yet, but he expanded this idea again with metabotropic receptors, but not with ligand-gated ion channels. In retrospect, I am surprised that, as far as I know, and I looked closely, nobody ever tried to expand this idea to ligand-gated ion channels. I'm happy.
They might have. Sorry to interrupt. I think have they not done it? Have they not done it with serotonin and serotonin and LSD and?
Yes, but serotonin in humans acts at specific serotonin receptors, which are metabotropic.
What is the difference between, and things like LSD that also act at that? What is the difference between ligand-gated and maybe I'm not understanding? Sorry.
Oh, okay. So, yeah, I should have said that, explored that before. I'm sorry. There are different types of receptors. The two main ones are metabotropic. Let's suppose that we have this type of serotonin receptors in humans, okay? Serotonin binds to it. It causes a conformational change. This is the membrane. And inside the membrane, there's another conformational change that interact with enzymes downstream, and they cause something. That's a metabotropic receptor. Because
is that like rhodopsins?
Exactly. They're exactly like rhodopsin. They are very similar, I should say, to rhodopsin receptors. Okay. So there's not an ion channel involved like in the nicotinic acetylcholine receptor. Okay. So in humans, actually, there's several types. Humans is an interesting case because there's a lot of serotonin receptor types in humans, but only one is ion channel related. All the others, including the ones that is related to LSD and antidepressants, for example, serotonin selective reuptake inhibitors, are metabotropic in nature. Did I answer more or less your question?
Yes, thank you. That was great.
Okay, absolutely. So The vibrational theory of olfaction, first, it did not begin with Dr. Turin. It was proposed in the 1930s to account for certain, what people observed, that two identical molecules smell different, okay? Dr. Turin extended that with electron tunneling, essentially. This hypothesis, it desperately needs to be further explored. And it's controversial, but it's not a bad thing, because I don't know if you read my hypothesis paper, but I said something like, when two equally capable minds disagree on a scientific matter, that's bound to advance the field. Okay, that's bound to advance the field, because essentially it's a ferment for growth, as it were. Okay, I mean, respectful disagreement is great in science. That's the way to go. Now, couple of additional things. How can we explore this? With structural analogs, like the further experiments that we are going to do with nicotine analogs and whatnot, and correlating with spectral properties, like Omid is beginning to do in collaboration with us. Another aspect of that is to explore deuterium varieties of compounds. For example, a nicotine molecule with deuterium that substitute for the hydrogen atoms. That will do next to nothing to the specific electronic structures of the molecule, but that will change the vibrational properties of the molecule. What I'm trying to say is that chemical reactions are controlled by the electron cloud, okay? And deuterium changes, it's like the strings of a guitar. Okay, so we have a fine string of a guitar, and you get a high noise pitch. But the same length of a string of a guitar in a much thicker one, okay, increasing by the molecular weight by deuterium, it's a lower note. And that's kind of the best way that I know how to explain this type of things. The point is that we are going to see different vibrations. We should see different vibrations with deuterium, okay? And that may give us an indication of whether the electronic clouds are going to be interacting differently. I need to expand this much better. And again, I apologize for my lack of physics and mathematical rigor, because again, I'm not trained for that. We can expand behavioral studies using this type of compounds. We can do direct binding, and there's ways to do direct binding and compare that to any physiological states that we can induce in a compound. We can isolate the same receptor that we isolated from electric ray and measure whether they are opening or closing and whether they are binding or not. So we can use these two types of approaches to concurrently explore these type of things. An exciting development that appeared in about maybe 20-something years ago was the discovery of a compound called the acetylcholine binding protein, which is a soluble protein. It exists floating in the cytoplasm of certain organisms, and is virtually identical to the nicotinic acetylcholine receptor. It binds nicotine, it binds acetylcholine, it binds most of the normal ligands, but it's a soluble protein, which simplifies things immensely biochemically, as opposed to a membrane-bound protein, okay? And we can keep going from there. Then there are some other papers later on in 2015, there's one that tried to explain the vibrations of several sets of ligands against their activation, again, on second messenger receptors, metabotropic receptors, nothing on ligand-gated ion channels, which is what we are hoping for. What I'm proposing, essentially, and I already apologize for my lack of detail of these ideas, because of I'm learning to do this type of things. What I'm proposing is that vibrational states can contribute to the interaction of compounds in ligand-gated ion channels, presumably through electron tunneling. Okay, so we don't know yet if that's going to be proven true, but we are going to test it. We intend to test it. I want to thank, again, all of you for being so kind enough to stay in the room and hear to me talk. And the worm, and very especially the people in the lab, past, present, and future, including the people who work in the current iteration of the Cotinine and Nicotine paper. One of them is here with us virtually, Emily. So, and again, I want to thank all of you. Money-wise, the College of Science and Mathematics of my department, the NIH, and NSF, which at times have certain times have funded aspects of of our research. And with this, if you have any further questions, thank you again.
Okay. Yes. Thanks for the talk, Professor Pagán. So sorry for some of the technical issues we had this end. Maybe we can start with questions for people that are in this seminar room, and then we can extend it to the people who are online. Are there any questions?
We spoke about it a lot. Just a quick question from the introduction part. You mentioned about the planarium and the bioelectrical change. You change the bioelectrical environment and see what happens.
Yeah.
Can you elaborate on this? What does it exactly mean, changing the bioelectric condition?
Okay. So for that, we take a step back and explain a cellular concept that it's called the gap junction. So what I explain in neurotransmitter release and all these type of things, it's called chemical neurotransmission. But there's also electrical neurotransmission. In this sense, we have, imagine that these are two channels, okay? So they're hollow, two channels. This is one cell, this is another cell. A gap junction is the joining of these two channels that when activated, they immediately equilibrate the electrical conditions in between the two cells, okay? There's experiments by a colleague in Boston. He manipulates, for example, Cotinine the planarian, manipulates the electrical property of the planarian slides by blocking those gap junctions, by blocking the immediate electrical conductance between the two cells, and that causes, if we have a square of a worm, it grows a head here and a head here, like again, like the one that I showed you before. And very fine-tuned, depending on the concentration, he can actually make that change permanent, meaning that if they generate a two-headed worm bioelectrically, okay, we are not changing DNA. We are not mutating the DNA.
Yeah.
We can create a colony of worms that if you keep cutting them, they will regrow two heads consistently. We don't know exactly how that bioelectric pattern is stored in cells. In the same way, you can manipulate, again, by gap junction inhibitors, these electrical properties of the cells in planaria, at least. And there's, by extension, they are hypothesizing they may be a good, actually an excellent way for regenerative medicine. For example, I'm going to be a little graphic. A person gets their hand cut for whatever. The idea is that in theory, we could generate like a contraption that you can put it there, add certain compounds, and enhance regeneration of a hand. We cannot do that nowadays with anything, okay? But those are not my experiments. Those are experiments of my colleague, but they can be done with relative ease in planet. And again, the marvel of it is that the DNA is not changed, and that as a biologist is something like sacrilege for me. That you can change the phenotype, the appearance of an organism without changing DNA, and that's just an example. I can give you many more, and I can actually send you a review of those particular experiments that I did, that I published a few years ago.
Okay, thank you. There is one question in chat. Should I read it?
Yeah, you can read it if you have.
Okay, the question is the interaction of acetylcholine and tryptophan from a molecular docking analysis or ideal interaction?
The short answer is that I don't know. That they have done molecular docking experiments, but that varies with the specific type of receptor. The dogma, quote unquote, is again that aromatic residues will interact with the positive charge of the nitrogen in the acetylcholine molecule. But I haven't seen any detail. There may be out there, but I haven't seen detailed structural analysis or docking or anything like that.
Okay. Can I just have a follow-up question?
Absolutely.
Didn't understand that part that you mentioned, that the small yellow part of the disulfide bind. Apparently, this is something very common in biology, but I don't know that.
No, it's not very common in biology either. It's very rare, actually.
Oh.
Because disulfide bonds are bound. For example, if we have a peptide, a molecule, right? It can have a certain three-dimensional shape that induces their function. Most of the time, when you see those cysteine residues binding to each other, it's like doing something like this, okay? Here's a cysteine. This is another cysteine, 100 amino acids, and they are separated by that. Very rarely we see those vicinal disulfides in nature. Actually, one of the few cases that I know is the nicotinic receptor. I don't want to say it's exclusive of that because I really don't know, but it's the only place that if you break that disulfide bond, even like this in vicinal, the receptor doesn't work. We don't know exactly why. It's fascinating, but the sad thing that is frustrating as well because we don't understand how it happens.
So it's necessary for the binding, for both nicotine and for both acetylcholine?
For sure, for acetylcholine. I don't know exactly for nicotine, because they have done experiments in which they break that disulfide bond by chemical means, and the receptor becomes inactive. But those experiments I have seen in the case of acetylcholine, presumably for nicotine as well, but I just don't know.
Okay, if anybody has a question in the online audience, then feel free to just unmute yourself.
Thanks. I have one last question. I was just wondering if anyone has looked at regeneration experiments with nicotine and cotinine, or have you?
We are doing that right now, very preliminarily.
Okay. But you haven't seen any.
But we haven't seen. I wish I could give you some results yet, but
okay.
We are beginning to. But I can give you an extension of that question. Of that answer. There's a series of pesticides, actual pesticides that are used in agriculture, that are derived from nicotine. They are collectively called nicotinoids. And they are a big environmental problem, because when they, for example, they spray those pesticides in a field, okay, it rains and it goes to a river, a lake, and it begins to kill non-target animals like fish, crayfish, flatworms, all these type of things. We are beginning to study those in a certain context, okay? I'm betting that cotinine and related compounds will alleviate the toxicity of those nicotinoids. But right now I can tell you that nicotinoids act at the level of the brain of the planarian. Why do we know that? These are completely unpublished experiments that we are furiously trying to write right now and expand it, okay? You remember the twitching that I showed you before, that some compounds, including nicotine, at high concentrations make the planarian twitch, all right? So we know from previous experiments, we're talking about 2013, about 11 years ago, that if you cut the heads of planarians, okay, the body of the worm keeps twitching with nicotine, okay? It doesn't care. That tells us that there are nicotine receptors distributed throughout the body of the planarian, which is reminiscent of what we see in humans. We good?
Yeah.
Okay. In contrast, if you add cocaine to those worms, right, they begin twitching, but if you cut their heads up, they stop, they don't twitch in the presence of cocaine. That tells us, at the time, and we actually got a publication of that. We actually got the cover of the journal. I can show it to you. That an intact brain is necessary for cocaine to interact, but not nicotine. Inspired by that, we're talking about a week ago. Okay, I was alone in the lab because this is summer school right now, so I'm teaching summer school. But we are not a research institution. I'm alone in the building. Okay, I was writing stuff and whatnot. I said, it's been a while since I've done an experiment. I want to do an experiment myself. And I thought about this idea, and I went ahead, decapitated a bunch of planarians, set a bunch of planarians of controls, and I added one of the pesticides. The intact planarians twitched like crazy. The decapitated planarians did not. That particular pesticide seems to act at the level of the brain in planarians. We have to do more experiments like that. That's a difference already between that nicotinoid pesticide and nicotine structurally. We can begin to explore that. And in my laboratory right now, I have maybe like 6 or 7 analogs of those nicotinoids, including a pair of chemicals that I bought of the nicotinoid and a deuterium-derived nicotinoid. I'm so eager to test that in the context of the hypothesis that we're doing. Okay, and again, this is non-published and everything, so please don't quote me. I don't know if it's going to be true or not. Right. It's just, but it's super exciting because we are testing the idea. Yeah, I'm telling you. And if you want, you can shoot me an email. I can send you the papers that we have done. Let me put in the chat my email.
Thank you so much.
Absolutely. Because I love doing this, and again, it's let's see. Oh, I just sent it to Emily. Sorry, Emily knows me. Okay, it's not letting me send. Okay, I sent it already. Okay, and I'll be happy to send you the preprints. We have a preprint of another idea, and we are studying the interaction of the compound that I have tattooed in my arm with cocaine, and we are doing the type of experiments that we are telling you, but with this other compound. So this is the best job in the world, okay? They expect me to read about science. Thank you, Mamaru. Did I say your name right? I hope I did. Okay, so the best job in the world. I'm expected to read about science. I'm expected to talk about science. I get to do research, and the students have to listen to me. So it's I'm enthusiastic about this. This is the difference between a job and a calling or a career. And I thank you again for your kindness, for enduring the biology talk, for Omid inviting me to here. And I hope that this is the beginning of a really fruitful collaboration. If you want to send for me to go to South Africa, I'll hop in the plane. I promise. Okay, and I thank you again for your kindness.
Okay, yes, thank you again, Professor Pagán, for the very interesting talk.
So can I add this last question? Because you mentioned that you are doing the regenerating experiment as well, but you are also not changing the DNA sequence, no?
No. We don't do molecular biology. No.
Okay.
We are manipulating. We are trying. I don't know if it's going to happen. We are trying to manipulate the regeneration phenotype, how the end result, without manipulating DNA. And that's an idea that's becoming into fashion relatively recent. And it's taking the biology worm. Oh, thank you, Bethany. By storm. It's I mean, it's these are exciting times for science.
Yeah, thank you. Thank you again. Sorry.
Okay, thanks. So yes.
Okay. So I'll be more than happy to send emails, preprints, reprints. So thank you so much, and I hope you have a great evening.
Thanks, you too, as well. Thanks for your time, and thanks for all the people attending online as well.
Thank you so much.
Thanks. All right.